Chris, thanks very much. And thanks, Brian, for the invitation to come and speak to you today. So first up, I go by Joss. Only my mother calls me Jocelyn. So Joss is good. First, a disclaimer. I didn't come up with the title of this. This was Brian. He put it on the schedule. I don't know about you, but when I see something 101, I kind of get PTSD and I get a flashback to high school. So most importantly, there will be no test, okay? But I'd be very happy to ask questions and through the poll, or if you want to stick your hand up at any time, go ahead. So we've all been in that place where you're sitting next to the guy on the plane and he says, "What do you do?" And you have your answer, your elevator pitch, right? So I say, "I study cuticles." And you get different kinds of reactions to that, right? So non-scientists often think about that. If you maybe read a bit of Scientific American, you like insects, you'll also, Google thinks that's a cuticle, okay? Here's what I think I do. So I'm a plant biologist. And I will then break out this sentence. Well, plant cuticles are specialized water-resistant cell walls that cover the surface of above-ground plants of all organelles. So they're plant skins. So I study the skins of plants, okay? And I do that work at Cornell University. Some of you may have been there, may know people from there. We're in beautiful upstate New York, at the end of the Finger Lakes. The main campus is in Ithaca. We have our Ag Experiment Station up in Geneva at the Agritech campus. And we also have the Hort Research Experiment Station, LIREC, down at the tip of Long Island. But my lab is on the beautiful green campus there. You can see it's just one of those main buildings in the middle, the plant science building. Been there for about, well, too many years now, a lot of years. It's beautiful, it's green, things grow very well. Sometimes. So in other parts of the year, when I leave this time of year, it's something more like this. But there's still things, and they're still green, and they're still growing. And then you arrive in Vegas. And things are still green and things are still growing, right? So life on land is very challenging, it's tough, it's hard to be a plant. But plants have managed to adapt and colonize every single habitat on Earth. They survive and they've evolved special processes to do that. And I'm really fascinated in my research and to understand how that is, to take that information and then take it out to people where it's useful. And so I'm funded a lot by the USDA, and they absolutely love it. We take our research and we translate it and talk to growers and breeders. And that's a lot of my job also as director of the School of Integrated Plant Science. So we have lots of research, but it's translated, and we have a lot of extension activities as well. So very broadly speaking, I'm very interested in polymers and structures that plants have evolved to allow them to colonize land. So all sorts of sizes and shapes and structures. They're very diverse. And I'm going to focus today for this presentation on this structure, the cuticle. Plant surfaces are important for many reasons on land. So perhaps primarily they're the barrier for water loss. They prevent plants from getting desiccated. But also uptake. When it rains, plants don't want to absorb water, right? So they create and set up water balance. They provide that hydrophobic barrier. They're critical in resistance to disease. They provide many aspects of environmental protection. And they're self-cleaning. And you've seen the lotus effect, right, where water beads up and rolls off leaves. And that will carry with it fungal spores and dirt and keeps that leaf surface nice and clean so you have maximum photosynthesis. I want to make one point. So I'll talk a lot about fruit today, but as I said at the beginning, all plant organs above the ground are covered in a cuticle. It's ubiquitous, it's everywhere. And so a lot of the principles and things we study, while we use tomato fruit as a model system, apply equally to stems and leaves and flowers and every aspect above the ground. There's even actually a very thin cuticle on roots. So we use tomato fruit. I learned to say tomato and not tomato after about 10 years living in the US. Tomato fruit for our research. It's very thick. It's very easy to get hold of. So we've all peeled tomatoes, right? And we study its biochemical composition, how the different components are linked into very, very intricate architectures. And then during growth and development, how that cuticle is remodeled. So it's not just a case of making it and that's it. There's restructuring all the time. It's constantly being assembled and disassembled and remodeled. And I'll talk about that in a bit. But to do that, to have that remodeling, it's a real engineering challenge. So there's a fruit, and constantly. It's having to deal with preventing water loss or uptake, fighting microbes that are constantly sitting on the surface and trying to get in. That's a big old bag of sugar, and those microbes want to eat that sugar. And during fruit growth and development, it's very dynamic. That goes from a small fruit to a large fruit, and that surface must remain intact as that organ expands. So it's going to be chemically very tough and impermeable, but it's also got to be elastic and plastic. It must grow and expand. And so there's an element that we're very interested in that dynamism, and that's the idea of self-repair. That as the fruit gets damaged, there are issues, cracking, plants have figured out how to heal those wounds. And what does that mean? How can we take advantage of that? And you've seen these sorts of things. So you can sort of put a wrapper on that and say, how do plants make, modify, and maintain their protective skins and cuticles? And you can see there, throughout growth and development, they do that generally very successfully. So there's a huge increase in volume, and most plants, most of the time, do that extremely well. So we're interested to know how they do that. But then, of course, sometimes they don't. And so in that case, we want to understand why, and is there something we can do about it? So we're making progress on a number of fronts there. So I'm going to jump back a few years and how I got involved in this business in the first place. I grew up in England, and I was working for a biotech company in the south of England. And at that point, they were very interested in using genetic engineering to improve tomato fruits. And over the last 20 or 30 years, the genes and the proteins and the metabolites that are the basis of all these key traits, color, flavor, aroma, have all been figured out. We know a lot now about why fruits smell why they do, the aroma components, why they taste the way they do, and what makes the color. To the point now where you can tinker with those genes, or you can regulate or breed for them, and you can generate very specific flavors and aromas and textures. So we have a really good handle on measuring those kinds of things and turning them about. The odd one out in this, however, is texture. It's really hard to figure out what texture is. It's not one pathway, it's many, many, many things. And so the ways in which we evaluate texture are also pretty crude and simple compared with the analytical techniques for these. Ever since probably before the 80s, there's this dogma that fruits soften. Texture is because of the primary cell walls. There's a little model of a cell, and you can see the structures, and we have some tools to try and identify which parts of the cell are there. What are the cell wall polysaccharides? So these are the long, complex sugars. And we even have very simple models of how cell walls might look. So we know, very broadly speaking, that the cell walls are made of cellulose, these long microfibrils in sort of beige there. These are cross-linked by hemicelluloses, this kind of barbed wire stuff, and then embedded in a gel of pectin. And through a lot of observations in the 70s and 60s, a model came up where fruits softened, this was the dogma, fruits softened because an enzyme called polygalacturonase, or PG, was made at the beginning of ripening, and it bound to pectins and chopped it up into pieces. Polygalacturonase breaks down pectins. And so the dogma was the pectins broke down, the cell wall fell apart, the cells separate, and fruit softened. So in the early 1990s, genetic engineering came up with ways of turning genes off. And so the idea was, let's just turn PG off, we'll stop softening. And so I came into the research environment just at that time when that was happening. And the very first transgenic crop came out. That was the Flavor Saver tomato, which some of you may remember. So everyone was very excited. Little problem, it didn't work. You turned PG off, it was gone, but the fruit still softened normally. So you got normal degradation, normal reduction in cell adhesion. You got a slight change in paste viscosity, but it didn't stop softening. So my PhD was looking at trying to understand, so why do fruits soften? Are there other kinds of enzymes that are responsible? Let's try knocking those out. And did that for a long time. And a huge amount of money has been spent, billions, on trying to tackle this at the biotech level, and we still can't stop softening. And I've been looking at that problem for a long time. Then, once I set my lab up at Cornell, we had what I call a game changer. So we came across a mutant, a naturally occurring mutant of tomato called delayed fruit deterioration. We named it that, DFD. This was a kind of tomato that it grew and ripened normally. There's a normal variety, it's a salad tomato called Ailsa Craig. DFD looked like a normal tomato, but it had a major difference. If you leave that tomato, pick it, and stick it on the kitchen counter. After a month or so, this will have softened, DFD hasn't changed. And you can even leave those fruits for, in this case, 9 months. You cut them in half, they're still juicy and somewhat fresh inside. They taste terrible. You don't want to eat a 9-month-old tomato because all the sugar's gone, it's turned to acids. But you don't need 9 months, right, at a commercial level. So we thought we'd found the holy grail. That's it. We've solved softening. Well, look at the cell walls of DFD. Look at what's different, and that will tell us why they're not softening. I'm going to just show this picture because it, I think, tells 1,000 words. This is just a time-lapse photograph of these tomatoes. Left and right are normal. That's the DFD in the middle. So here's the holidays going ahead. And you can see these are desiccating, drying up. This guy starts to try and escape. DFD, not moving. So it's a really dramatic difference. So we looked at the cell walls, and they broke down and degraded just the same as normal tomato. So we were completely stumped. But then we noticed, we thought about this more carefully. We thought about disease resistance. And normally if you leave tomatoes out, they get infected by microbes. DFD never does. So we did this experiment, and we took 2 commercial varieties, Ailsa Cragen and something from the supermarket, and DFD, and we punctured it with a fungal inoculum. They all got infected at the same rate. But if you do this if you put this fungal spore solution on the surface, these get infected, DFD doesn't. So that was the lightning bolt. We thought, it's all in the skin. We've been looking at the wrong thing all along. So that made a lot of sense, right? The DFD fruits stay firm. They're not losing water as much as the wild type. We measured that. And so water doesn't get out and pathogen doesn't get in. So there was a kind of giant slam on the brakes. And we said to the lab, right, we're not looking at polysaccharide cell walls anymore. We're going to look at skins. So that's what we did. We started changing the focus of the lab onto cuticles. So what do we know about cuticles? So multifunctional structures, there's a little diagram there of the cuticle sitting on top of the polysaccharide cell wall, and there's a couple of cells with a cytosol and vacuole there. Many, many functions, right, have been evaluated over the years. And a number of groups have started to assign functions for the different components. We know that the cuticle has a range of waxes, and they contribute very much effectively as a water barrier, and they are effective for pathogens and this self-cleaning surface. As well as that, there are some phenolic compounds which act as very effective UV filters, right? So these will absorb the light and prevent the UV getting in and damaging the cells inside. And then a major part of the cuticle is this compound called cutin. It's a very complex structure. We'll come to that in a minute. But essentially what we're looking at is a combination of these components made in some way. So what do we know about that? So we said, okay, right lab, let's look at the literature. What do we know about cuticles in fruit? Let's look at that and start to get back to our DFD. So this shows a cross-section from the surface of a fruit. This is inside the cell. There's the polysaccharide cell wall with the cellulose and hemicellulose and pectins. And this is the big cuticle on top of that. And we looked at the literature. And over a couple of decades, a lot had been learnt about the synthesis and modification of that polysaccharide component. We know a lot about the bells and whistles and dials and knobs. Cuticle, far less. So we looked, at that time we started, almost nothing was known about the biosynthesis or how cuticles were formed. So that's really been the focus of our lab for a couple of decades now. What do we know? So this shows a sort of cartoon we put together. So we can think about, there's the polysaccharide cell wall, there's the cuticle, and we can think about these components of cutin, that comprises all this stuff in the middle. And then there are waxes. These waxes can sometimes sit on the surface where they form as crystals. So you've seen that, the gray film on grapes, for example. Or they are embedded inside the cuticle as intracuticular wax. So what functions are that? And we can think about this in a different way. We can think about the waxes sitting on the top, and again, as this really important water barrier, almost like a sealant on top, right, preventing the water from getting out. We can think about the cutin as a scaffold within which you deposit all the waxes that sits in there, almost like a sponge, right? And then that's enmeshed with all of the underlying cell wall polysaccharides. It's almost like rebar, right? There's this rebar, and then there's a cuticle on top. And they provide a very important structural component. So this is, again, a very, very simplistic cartoon, and the more we look at it, the more complex it is. But I think this gives a good idea of how those functions operate relative to each other. So cuticle waxes, cutin, and cell wall polysaccharides. So we have a lot of questions, right? How do those different wax components affect permeability? And some waxes pack very tightly to make a very crystalline, impermeable structure, and water doesn't pass through very easily. But other kinds of cuticles, you'll see other waxes that break up that crystalline structure. They're not so linear, so you get a path that water can pass through more easily. So in other words, it's the combinations of different waxes will assemble in different ways and create different degrees of water barriers within those cuticles. We're also very interested in how cutin, that scaffold, is made. We made some progress there. And then what's really important also is how is that cuticle attached to the polysaccharide cell wall that's underneath? Why is it that you can't just pull the skin off by itself? You always rip off the cell wall as well. So it's the combination of those things, I think, that are really big mysteries. So I'm just going to quickly talk about some of the technologies we've used to look at these questions. So we've been developing techniques for high-resolution imaging of cuticle architecture. It's important if we want to get those knobs and whistles and dials on the cuticle to find out what are some of the genes that are present in that one cell layer that makes the cuticle, and also some of the proteins. And then we use genetics, looking at mutants and engineering, to see if we can test those theories and find out, can we control how those cuticles are made and optimize them? And we know, for example, that some of the wild species of tomato live in really dry, desiccating environments in the Andes, and they make fruits that don't desiccate. So can we take advantage of those structures and introduce those into domesticated cultivars? So, looking at imaging, when I arrived at Cornell, there was a guy who was about 190 years old that was just retiring, and he gave me this sort of box and said, "Oh, you want to work on cuticles? You should use these." And it was like a sort of Harry Potter box of little brown vials with copper plate writing on. So, all right, we can try that. So my student got working on this and stained cuticles with all sorts of these funky things and came up with some amazing pictures. So we get different contrasting dyes. Some give nice contrast, some show this kind of dichromatic pattern, so you can show different structures appearing. But there was a really interesting class of some of these dyes that were fluorescent. And you can take advantage of fluorescence now with high-resolution microscopy to get three-dimensional images. So this shows a typical cuticle sitting on the epidermis of tomato fruit, and you see it's big and thick and it's complicated. And it's very hard, though, to understand the three-dimensional structure of those things, right? You can take lots of sections, but you can't really see how it's all put together. So if you really want to look at cuticle function and deposition, you really want 3D. So that's what we did. And we did three-dimensional imaging with these fluorescent cuticles. And you can start to see now really high-resolution pictures of what the cuticle looks like. And we played around, and you can even get this sort of George Lucas Star Wars version of the cuticle, right? You can zoom about all over the place. So by using these kinds of technologies, we can really understand how the cuticle is developed and how it responds to environmental stress and how dynamic it is. I'll stop, they're going to get seasick. All right. So that's in tomato, and we did a similar thing. And the key point here is that all organs have cuticles. If you look at different kinds of fruits, you see an enormous range of structures, okay? Different ways they're put together in different patterns. We don't know why that is. We have no idea. Why do you have a different kind of cuticle in a nectarine compared with a huge, thick cuticle of a mango, right? But by documenting this and looking at this and looking at how dynamic it is, that will start to give us some clues as to how to investigate that. But the same thing applies also to leaves and stems and flowers, right? They all have cuticles. On leaves and stems, typically you have much thinner linear cuticles. So we have now an ability to look at the architecture of these things. How do we get to the genes that make them? How do we understand those genes that are expressed just at that one cell layer? A lot of people have been looking at genes and when they're expressed in fruits for a long, long time, decades. And what everyone does is they get their tomato and they get it out from the lab and they chop it up, and they put it in liquid nitrogen and they powder it and they extract things from it, whether it's metabolites or proteins. And the problem for our perspective is that we don't care about most of what's in it. There's a box set that shows this bit, it's a cross-section. We don't care about most of that. All we care about are the genes and proteins that are expressed in that one cell layer that make the cuticle. So by grinding everything up, we've always had serious dilution effects. It's very hard to detect those genes, and we lose all this critical spatial information. So how do we understand what techniques can we use just to look at that one cell layer, ignore everything else? So we use a technology called laser microdissection. And this essentially is taking slices of fruit, and then you put it under a microscope, and then use a mouse, and you essentially just draw around the area that you're interested in. In this case, this is the one cell layer that makes the cuticle, makes all this cuticle. That's all we care about. And then you can hit a button and a laser will be fired and trace around those cells and then pop that material out into a tube. Then you take that out and extract the information you need to look at which genes are expressed. Or you can look at proteins or cell wall polymers. So what we did was we had a big project looking at this, using this technology, looking at the different cell types of tomato, so comparing that with that, and immediately things that are critical for making cuticles jumped out. Those genes were expressed in these cells, but not in these cells. That shows an image here of what happens after we've been slicing and dicing. So you can see there's a gap missing for that cell type, there's a gap missing for that cell type. We looked at this vascular tissue here, and so on and so forth. So this gives a really nice way of very surgically understanding what processes are occurring just in certain cell types. We made a database about that, and you can see now we can start to paint on just genes that are expressed in the outer epidermis and ignore some of those other things. And we're interlacing now with two-dimensional images to see which genes have particular cell and tissue expression patterns. And then we also want to put that onto a 3D database, and we used a technology called computed tomography, where we can use basically soft X-rays, and you can go to a website now and you can grab the slider and drag it across. And that gives you the ability to look inside the fruit from pretty much any direction. And you can measure aspects of it, how much locular jelly there is, what do the structures look like. You can click on, in this case, the other direction and pan up and down. So now we can digitize our fruits. So we can know which genes are expressed exactly when and where, and we have a digital tomato fruit. And we're doing that in 4 dimensions because we're looking through time. So the idea is we want to have the life of a tomato from birth to death, right, till it's fully ripe, with every single gene that's expressed, and understanding when is that cuticle made and how is it remodeled. That's looking at genes. We also have looking at the proteins, the enzymes that make it. This was more of a brute force approach. There's no cool technology here. This was me sending out my PhD student out to the field and said, go bring me 50 kilograms of tomatoes. So he looked at me for a while, then he went and did it. So he took every single one of those fruits and got a little metal wire and dipped it in a little spinning vial of methanol and chloroform. And what that will do is that will dissolve all the wax, the cuticle, off the surface. And he did with the next one and the next one for a long time. And after 50 kilos, he boiled that down to about 4 grams of wax from those fruits. I bought him some beers for doing this. and within that 4 grams of wax, he got out of those about 100 micrograms of protein, right? So he very carefully walked across the room and we took it to the mass spec. And you can use the mass spectrometry to say, what are the proteins? So out of that, he got over 200 distinct proteins, many of which we hypothesized were critically important for modifying the cuticle. And what was really cool was some of those proteins that he identified matched up with the genes that we had done through the laser dissection. So now we've got really good candidates. So what we can do now is then take those, we can turn their expression off or up and have a look at the effects on the fruits to reprove that's what we've done. So that's when you think about things carefully and plan, and you're really, really careful about what you do. You also, in life, need to be very lucky. Serendipity. So I'm going to tell you a bit of how we got a bit lucky. We were really interested in understanding the permeability question, right? Why do different fruits lose water and desiccate more than others? And we had a collaboration with a great guy called Danny Zamir, who runs a big tomato operation out in Israel, in Rehovot. He was growing in the field, I think it was several thousand different hybrids and varieties of tomato, anything he could get his hands on. And he was putting it out for the community. Come and have a look. Use it for whatever you want. I thought this would be a great way to look at understanding water loss. So I got another graduate student, Eric, and he went through this experiment of taking fruits from every single plant. And again, I had to buy him beers, weighing those fruits over essentially a couple of weeks, and just looking at how much they lost water. So he documented this. I think he liked it. But what came out of that was actually some really interesting data. So this is the only data set I'm going to show, okay? So it's a graph, apologize. So this shows 3 kinds of tomatoes, right? This like a persicum in gray, that is your tomato you buy in the supermarket, all right? So this is the number of plants, and this is the water loss rate. So bunch of tomatoes lost this around 2 milligrams per centimeter square per day. Some lost really fast, and some very little. This is another kind. This is more a wild variety, the Pimpernellifolium in blue. So the wild tomatoes lose far less. If you cross them in the middle, you get something in the middle. So we were interested in two things from this. We were interested in two sets. Why are you having fruits that are losing water really fast? And also, what's going on when you have fruits that lose water very slowly? Is it a cuticle thing? Well, with this, what he noticed was that, in fact, what you get, and you'll have seen this in some of your fruits, is you get these micro cracks, right? Often by the stem end. And what these macro cracks allow is lots of water loss very fast. So he used this stain TB, toluidine blue, and what that does is it penetrates. If it hits the cell wall underneath, it'll stain the cells very easily. It won't penetrate if there's a cuticle. So whenever you see blue staining, it means there's a crack and water's gone through, and the toluidine blue has gone through, okay? So he screened that, and any of the fruits that had these microfissures, we didn't incorporate in the experiment, because then the fruits were damaged. Going back to the data then, what was the key thing? What was it that determined water loss? And after a lot of analysis, we had a puzzle. The water loss was not related that we could match across all of these thousands of varieties to any wax profiles. Didn't match the amount or type of cutin, and it didn't match cuticle thickness or architecture. We couldn't match it with cuticles at all. But one thing Eric did notice was there was a completely strong correlation, a relationship, with the appearance of these dots on the surface of the fruit. Look like little bullet holes. So I was like, what are those? Appear to be some kind of pore. So we did some of our three-dimensional imaging, and I'll show you a movie now which shows a cross-section of the fruits. The cell walls in polysaccharides in blue, and then the cuticle is shown in this gold color at the top. So there's the hole, there's the bullet hole. You can see right down the middle, there's a pore coming straight out of the cuticle. So you have this structure on the top, and that creates a quick channel that all that water is coming out, and that's why the fruit is desiccating. So what are those? Well, if you remember, you've probably seen this on small green tomato fruits, that they're fuzzy, they're covered in hairs. And that, what you just saw, was what's left of a hair that's been knocked out. They're called trichomes, and they cover the surface of tomato fruits. They cover the leaves as well and stems, you'll have seen those. So this shows an image that you can see the trichome has been snapped off. When you snap that hair off, you leave a hole. So that was interesting for a start. Different cultivars have different numbers of trichomes and holes, and we can think about how to design fruits that don't have those. But what was also really exciting, we noticed, was if you wipe the surface, after 4 hours, in some cultivars, that hole was sealed up. So something's happening in the cuticle, it's repairing itself, which was completely surprising. So we're now focusing on that structure. So plant skins. To wrap up, structures of skins are highly complex, and I've just shown you a tip of the iceberg. We now have a lot of analytical tools to look at them, and we can start to piece together how these things are being made. So we've got a basic framework, but this really is the iceberg. There's huge unknowns, right? How the microstructure, ultrastructure varies, what each of those components are contributing in terms of barriers to disease or water loss. How do you assemble it? And then the translational potential, right? How do we take this information and use it in real-world settings? How do we use it for growers and breeders to actually make something that will make a crop that's more valuable? So I believe it's an extremely important target. We have the technologies to enhance cuticle structure and function, and I think that's really a theme here within this meeting. And I think you'll see from the next speakers, how they're using the products and thinking about cuticles to try and modify the quality and products of the crops that you guys are making. Okay, I won't go through everybody, just a cast of thousands. I'd like to thank everybody in my lab. Cornell is a great place to do tomato research. We have a lot of people working there on different aspects of tomatoes, but also collaborators all around the world. And I should acknowledge the funding for our lab comes from NSF and the USDA. Thanks very much.